So it is noon. To keep us on track, I'd like to get us started. So for today's Stages seminar, our trainees are going to be giving presentations about their research that they're doing or planning to do for their master's or doctoral work. So without further ado, our first presenter today is B. Devona.
Okay, so I am a fourth year PhD student in the MARB program. And so I'm just going to present some of my introductory dissertation slides. And so my dissertation is titled PFAS-induced immunotoxicity and later life immune consequences using Oryzes melastigma as a model. Okay, so for those of you that are not familiar with PFAS or PFAS, they're per-polyfluoral substances. They come from a variety of sources, so we're exposed to them daily. These are just some of the primary sources, so drinking water, fire extinguishing foams, food packaging, personal care products, and manufacturing and textiles. So there are tens of thousands of these compounds, but I'm obviously not going to look at all of them. So I am just looking at 6 of them. And 2 of them being the legacy compounds that are no longer produced in the industry, but they're still present in the environment and still on a lot of products. And then the other 4 are replacements for those legacy compounds. And so they're just slightly shorter carbon chain compounds, but still sort of the same function in industry. So we would propose sort of the same concerns as far as immunotoxicity goes. And so I have some just bullet points from literature about what we've seen as far as immunotoxicity of these compounds, a lot of stress and alterations to the metabolism, changes in inflammation and oxidative stress, and then atrophy of the thymus and other key changes in immune tissues. So those are sort of things that I expect to see when I expose my fish. And so how this relates to human health, because my sort of interest is in relating environmental health and human health. We are concerned with the prenatal and postnatal PFAS exposure because this is the age in which there's immune development ongoing. And so alterations to immune development could then lead to consequences. And so there are levels of PFAS measured in blood of humans, especially pregnant mothers and early children. So again, those developmental windows for the immune system. These are just some of the levels that I found. There's a lot more newer stuff out there now. So this table needs to be updated because they're pretty much all present in human blood at some concentration. And so my overall arching hypothesis is that critical windows during development are susceptible to toxicant exposure, and that can lead to dysregulation and dysfunction of a system. For me, it will be the immune system that I'm looking at. And then even after maturation, there could still be later life impacts on those fish. And so I use marine Medaka in our lab. They're a model organism. We have their genome sequenced, so they're very good for looking at molecular endpoints as well as developmental stages. These are some of the immune developmental stages that have been previously outlined by my PI, Dr. Seaman. And so basically, I compare this to human fertilization. And so that is also the development of the immune system for humans. And so how that relates to my fish. And then I choose age groups within that to test and to expose to my PFAS. And again, they're just a good model organism for various reasons, mostly because they're also available to us in our lab. And so my overall hypothesis is that PFAS exposure during immune development will have direct and long-term impacts. So the first step would be to identify critical windows in the development of these marine Medaka, and then to assess the differences of PFAS exposure. So short chain, long chain, different functional groups and things like that. And so I'll compare the critical windows, different types of endpoints that I'm going to look at, and then immediate and later life, and then again, active or not active immune system. So are they stimulated by stress or a pathogen or not? And so my first chapter was to identify those critical windows during immune development. And so again, this is what we know already based on my PI's research of cellular changes in immune development. And so I wanted to look at changes in immune competence, which we define by the ability of the fish to fight off a pathogen, and then also changes in immune gene expression. So during immune development, different genes are turned on and off at different times to develop that immune system. And so I wanted to look at those, when those changes are happening, because those may be more susceptible periods of time. And so I hypothesized that the previously described windows would be incorporated into my newly defined critical windows. So to do this, I took different age groups. I took 20 different age groups between that period of time in which development is occurring for the immune system, and I exposed them to a pathogen that I know induces an immune response in our marine Medaka. And I did this, and then I monitored their survival essentially. And then I also did this and then collected them after 48 hours of exposure, so stimulating their immune system. And then I monitored their genes on and off with an activated immune system to see where those changes in gene expression might be occurring. And so these are the different types of genes that I measured, if anybody was particularly interested in that. I tried to measure lots of different types, but a lot pertaining to the innate immune system, which is like fully functional in our marine Medaka biome. And so my conclusions, I'm not going to go through all the data because that's just like so much, but I found 3 critical windows, and they did encompass those previously outlined cellular developmental windows. And so my next step is to then see if these are more susceptible to the PFAS exposure. And so I will be exposing my fish during these 3 critical windows, just for that duration of time, and then seeing what happens to them afterwards. So again, I did that first part, identification of the critical windows, and now I'm on the assessment of the PFAS immunotoxicity, which is taking me a while, as you can imagine. And so again, I'm addressing that hypothesis that exposure to a toxicant during just that period can lead to dysregulation and dysfunction. I'm using concentrations relative to people's concentrations of PFAS in their blood, specifically those pregnant mothers and early childhood children. And then I'm also using EE2 or ethinyl estradiol as a positive control, because we know that this induces consequences to the immune system during development. And again, I'm just trying to identify PFAS that do induce some sort of response, and at what concentration. I have a high and a low dose, but both still very relevant. And then I suspect that they will all be equally susceptible, that there won't be a big difference between the PFAS or types and or concentrations. And that earlier critical windows might be more susceptible, but that ends up not being true. So it's a hypothesis. And so I sort of have the same sort of setup for my exposure. Each individual is exposed to the PFAS separately, so they're getting sort of a known dose in my mind. I'm only again using one of those concentrations and one of those PFAS, so not getting sort of any intended mixed exposure. And then my control is just no intended exposure. And so then I'm looking at the immune competence. So again, stressing them with a bacteria and assessing how long it takes them to sort of succumb to that pathogen. And then looking at their histology, so focusing on those immune tissues to see if there's any effects. And then looking at the transcriptome. So how are the genes and the regulation of the genes affected by exposure during development? I sort of finished this, but not completely with the transcriptomics. And so I wanted to rank my PFAS in comparison with EE2, which they were all not as bad as EE2, I should say, but we kind of expected that. Obviously, that's why we used it as our positive control. And then looking at the tissue level, I haven't gone through all of my histology, but I am trying to see essentially if the thymus volume is lower. That's pretty much what I can measure at that level. And then for the molecular level, I'm looking at the transcriptome, and we do see differences in gene expression, but I have not characterized those genes just yet. So I'm not sure which pathways are more affected just yet, but I expect things that I see in literature, which are pathways associated with the immune system and fighting off pathogens. And so then my chapter 3 is raising them to adulthood after that initial developmental exposure. And so same sort of idea, except I won't assess anything until after they're sexually mature. So that's 3 or 4 months old. So I'll still just expose them for that few days of their embryonic or larval stage, and then raise them like I would just a normal fish in our lab. And I do expect them to still have some sort of negative outcome. If I saw initial negative or direct negative effects of PFAS, I expect that long-term those would persist. And so same setup of exposure, except I raised them to 2, but probably more like 3 or 4 months Once they start producing eggs and mating, then I'll call them completely mature because I can differentiate between males and females at that point. And then I will assess pretty much all the same things, except at this point, I can do a few more cellular levels because we have more tissues. And I expect to be able to compare them directly and long term. And again, these are just some things that literature has seen. And so I expect to sort of see some of the same outcomes. And altogether, I'm hoping to like sort of bridge the developmental and toxicity relationship. That's important for like long-term assessments and risk assessment. And then, yeah, this is everybody.
Time for a question, if anybody has a quick question.
Yes.
So I think you chose to stick to the best representative, what's left over in the environment. But when I think about where this is going in the future, and you have Chemours over there, right? And it's the world leader in the replacement refrigerant, which is short chain. So do you think maybe adding one of these replacement?
I'm probably not at the point where I can add anything at this point, but because I'm like already raising them to adulthood, so I'm kind of just like waiting at this point. But yeah, I mean, I picked my PFAS based on what I could find in literature because I was trying to like sort of justify why I was picking them. And there wasn't a lot out there at the time back in like 2020 when I was like looking, and now there's like all this stuff is just coming out about all these different short chain compounds. And so probably I mean, I'm hoping to test the water and see what I've got in the water to see what I'm exposing them to accidentally, not really accidentally, but non-intentionally. But yeah, but I mean, there are definitely other ones you could be looking at, which probably have very similar effects.
Yeah, because I think the short chain, the idea is it's not going to bioaccumulate, it's going to
Yeah.
break down, which I don't think anybody does.
I'm not sure if that's really what's happening,
Right.
but
And real quick, while we get the next slide set up, I would like to invite our stakeholders and community partners and our faculty that are joining us today to give quick introductions. So your name, either what your lab is called or who you're representing today.
Outside of the city limits?
Hmm?
Oh,
Yes, so James,
Jim Tolan with Texas Parks and Wildlife, fisheries biologist.
Patrick,
Patrick Larkin with Department of Physical and Environmental Sciences.
Hussein,
Hussein Abdullah, Ben's department, organic biogeochemist.
Dave Felix, Penn's Department, Chemistry.
Mary Kramer, Coastal Bend Basin Estuaries Program.
and then at our next break, we'll do the same with those joining us remotely. So our next presenter is Amanda Burton.
Hello, everyone. My name is Amanda Burton, and today I'll be talking about alkalinity in Texas bays and its relationship to seagrass genetic diversity. So this is a little overview of what we're going over. I'll talk about who I work under, some general things you'll need to know so you can stick with me through this, what I'll be doing, and some previous studies of alkalinity in seagrass. So I'll be working under Dr. Patrick Larkin. He is my graduate advisor. He works in plant biology, genetics, conservation. He's a chemistry professor, and we'll be working closely under Dr. Xingping Hu, who works at the Heart Research Institute. He does many different things, but for the purpose of my research, the most relevant is chemical oceanography. So why is seagrass so important? Seagrass is kind of the forgotten coastal environment. It's not as pretty as coral, and you can't see it as easy as mangroves, but they're incredibly important. They are nursing grounds and habitat and food source for many fish species. They protect the coastline by holding sediments in place and counteracting erosion, as well as dissipating wave energy. And they are a mitigator for climate change and thought to be a significant carbon sink. So why is alkalinity so important? Alkalinity in seawater refers to its ability to resist acidification. It's often confused with basicity, and they're related but not the same. The rebel factor is the water's ability to absorb CO2 from the water, so that has an inverse relationship with alkalinity. And ocean acidification, it has many negative effects, but most prevalent in what I'm talking about is going to be marine osteoporosis, which I'll talk about in a second. And bays along the Gulf of Mexico are becoming more acidic. So what affects alkalinity? Acidity, obviously, is going to be a big effect for that. Freshwater inflow of water as well as sediments. So this picture on the right is sediment input from the Mississippi River into the Gulf of Mexico. So that just shows you how much that part of the carbon system is important. And then evaporation rates affect it, as well as calcification reactions. So this is a calcification reaction. It's a natural occurring thing. It's not a bad thing. CO2 mixes with water, turns into carbonic acid, and then it later dissipates into hydrogen and carbonate ions, and then connects with calcium, and benthic creatures make their calcium carbonate shells from this. But when it's already acidic, like you don't have enough primary production, you have too much CO2 in the air, as well as many other things, the water becomes undersaturated with calcium carbonate. So in layman's terms, the water holds on to the calcium carbonate, so there's not as many ions to make shells. So these organisms either don't make shells, or they do, and they're just formed like the red one on the right. So I will be looking at seagrass genetic diversity in Halodili radii with Dr. Larkin and Dr. Hu. Halodili radii is the most abundant seagrass in Texas by far. So it's a good one to use for a pilot study, because if there's seagrass there, there's probably a lot of Halodili radii, and you can do good statistical analysis with that. In theory, the more genetically diverse a plot is, the more stable its conditions are. So Coggan et al. Did a study in 2023 where they talked about using seagrass as an indicator for ecological disturbances, such as hurricanes and droughts and whatnot. They found that Texas seagrass beds are decreasing at a rate from 1 to 7% a year, and southern Texas estuaries are prone to be hypersaline. And so anything that is anthropogenic that could make that worse, such as building a dam, or if there's lack of rainfall, that'll just get even worse because it's already prone to being hypersaline. So they used seagrass. They said seagrass would be a good ecological indicator because it's long-lived and clonal. So it's easier to research that than it would be to say, try to look at alkalinity based off of seashells, because first you have to find the seashells, and then you have to analyze, and then you have to rely on a model to tell you what it represents. So seagrass is just a better thing to look at here. And they found that the most important factors that were related to disturbances were percent cover, seagrass species composition and diversity, which I'll be subbing for genetic, shoot allometry, which is changes in length and width of the blades, and stable isotope ratios. So Dr. Hu, the same one I mentioned earlier, he did a study in 2015 where he looked at 4 decades of alkalinity in the northwestern Gulf of Mexico, and he found that many of the bays are becoming more acidic. You know, surprise, surprise. He hypothesized that lack of rainfall and building of dams is a big contributor to this, and he suggested that better water management would help. So Zeller et al. Looked at some seagrass meadows in the Florida Keys, and this one really showed the importance of that rebel factor, where total alkalinity production was a sink for carbon dioxide when the total alkalinity was higher than the dissolved inorganic carbon that was introduced by other means, including from the sediments. So the site ended up being a carbon source, not a carbon sink, but it would have been much more of a source if there wasn't already the reactions that mitigate it. It just wasn't what you would think. Miller and Kelly looked at seagrass in Alaska, and once again, they found something that you wouldn't think. They were looking for phytoremediation of seagrass, and phytoremediation is when you plant something in the hopes to mitigate a toxin. And so they found that actually a really dense seagrass bed does not mitigate CO2. It doesn't sink it as much as you think it would, because at night, when they're not photosynthesizing, they're still respiring. So they found that actually the best patch to reduce alkalinity and sink CO2 had patchy seagrass, but had microalgae present. So brief summary. I'll be studying alkalinity as it relates to seagrass genetic diversity in Texas bays under Dr. Larkin and Dr. Hu. In theory, the more diverse, the more stable the habitat. Alkalinity in seawater is its ability to resist acidification. And alkalinity is affected by freshwater and sediment input via rivers, evaporation rates, and calcification reactions. Seagrass meadows are considered a blue carbon sink, but it can be a source depending on that Rebel factor. And it's possible that the best seagrass meadow is patchy with microalgae present. Any questions?
Any questions for Amanda?
Did you study the relation between the ship traffic and the seagrass in that area?
We have not, we aren't looking into that part of it yet. Right now, we haven't started yet, but we're gonna look at river input into the bays and do it based off of mostly freshwater input and sediment input from the rivers.
What was your study area gonna be? Sorry, I missed it probably.
Alkalinity as it relates to seagrass surface.
Study area.
Oh, chemistry.
Where are you? looking?
We're gonna be looking a lot at Capano Bay, Corpus Christi Bay, a lot of the southern bays in Texas.
Because if I recall correctly, Dr. Hu's study, there was a gradient, right, along the coast, and the level of acidification was not so bad going down south.
Yeah, south, he said northwest had the most acidification going on. We're still hashing out exactly which bays we're gonna do because we needed to find out how many seagrass meadows we need to do genetic analysis for to be important, and we're still hashing that part out, but it'll be southern Texas bays.
Okay, thank you, Amanda.
As we transition, I want to remind everyone that this is a time for you to give feedback to each other, even on presentation skills. If you don't want to do it here, maybe afterwards when you sit down and talk to each other, it's a great opportunity for peer feedback.
Okay, so while we get the next presentation set up, I'd like to invite those joining us remotely. If you're able to turn on your cameras and mics and just introduce yourselves really quick.
I'm Patrick Knight, Ingleside on the Bay Coastal Watch Association.
I'm Catherine Maston, also Ingleside on the Bay Coastal Watch Association.
I'm Lee Perry with the Coastal Bend Bays and Estuaries Program.
Aaron Baxter, Coastal Bend Bays and Estuaries Program.
Evan Turner with the Texas Water Development Board in Austin.
Lucy Flores with the Texas General Land Office, Texas Beach Watch.
Hey y'all, Jason Pinchback, Texas General Land Office.
Nick or Calusa?
Hey all, Nick Farmer, NOAA Fisheries Service.
Hi everybody, Calusa Horn, NOAA Fisheries, Protected Resources Division.
Okay, thank you. And our next presenter is Laura Button.
Hi everyone, I'm Laura Button. I am a CMS's master's student, and Dr. Dorina Margula is my graduate advisor. So, forgot to say my title. So my title is Groundwater and Health Analysis in Rural Communities in Nueces and Aransas County. So it is going to be in Nueces and Aransas County, but more specifically, kind of in the outskirts of Aransas County here. And then in terms of Nueces County, everything unincorporated. So everything starting from Tierra Grande over here, Petronila, a little bit of a community down here called Golden Acres, going back up to Lost Creek, and then Enquete, everything over here in Cindy Park, and then in the 73 area. So the goal is to try to have as many wells spread out, so about 20 throughout that general area to monitor the water conditions going on. So what I think is the most important and unique part of the project for not just mine, but the overall thesis project that this is involved in is the community. It's the colonias and along the US-Mexico border. So they're typically composed of Hispanic, low-income people who live in these self-built homes, typically on agricultural land. They don't have proper drinking water. Usually sewage systems aren't the most developed, and they lack basic city services such as trash pickup. So if you've ever lived rural, you kind of know that if you don't have trash pickup, you either need to purchase it privately, you bury it, or you burn it. So you can already see how some environmental issues occur there. So this is a picture not that long ago, probably about a week ago in the Tierra Grande area. So that's right here. That's the closest one to us. It's probably only about a 30-minute drive away. So this is after just overnight rain. And that's about like 3 feet of water just going down because it's very low elevation. It fills up really quickly. And then this is a picture in Hidalgo County. So a big difference between the colonias that are going to be here and down by the border, which are Hidalgo County, Cameron and Willacy, are that there are laws protecting the colonias down there. So anything within 120 miles of the border is going to have some type of rule applied to it. In Oasis County is 150 miles. So it's a little more no man's land over here. But what's kind of a little even more unique about the communities here is the clay-like soil. So if you know clay-like soil, it's gonna shrink and swell. And then once you hit that infiltration rate, the water is just gonna pile on top like you see in that first photo. And then you have proximity to contamination sites. So if you don't have somewhere to dump your trash, there's lots of landfills near you, TCQ sites, and Superfund sites in those rural areas out there. Because you can just kind of a little bit do what you want once you're out of the city limits. And then you have shallow groundwater wells, which because we're at such a low elevation, once you hit water, you can just use it, and that's not safe either. And then drought and flood periods. Corpus Christi is just a little funny with the weather sometimes. A lot of these communities exist by tradition, and when I say tradition, it's very normal for them, but it's a cycle of poverty, lack of education, and all of the above. But what I'm hoping to do with my project is include the surveys. So we're going to have an extensive survey that includes factors like occupation, health issues, water usage and concerns for your water, and then a political outlook. I'm more concerned about the health issues and water usage because that'll definitely tie into what comes back from a monitoring well. Because a lot of people, for the most part, don't drink the water, but they use it on themselves. So dishes, showers, anything like that. But some people still do drink it. And then the well testing. So the idea is to put 20 wells in Oasis County, 10 wells in Aransas County. So from there, it'll get tested monthly or right after a rain event. And we'll test for things like metals, radon, radium, stable isotopes, and arsenic speciation. The funding for arsenic speciation isn't as much as the overall, so that won't happen as often, but it's still going to. And that'll tell us a little bit more about exactly what's going on. So and I'd like to focus on arsenic. That obviously depends on what comes back from the testing to begin with. But the two forms that will be tested with arsenic speciation is arsenate and arsenite, which are the inorganic forms. From there, with Hispanic communities in South Texas, diabetes is pretty prevalent, and I'm saying arsenic is the cause of it, but it would be pretty interesting to find out if there's a link to drinking your well water that is contaminated with arsenic and then having the possibility of diabetes or just being more prone to it. So as you can see here with arsenate or arsenite, they lead to insulin resistance and insulin secretion, hyperglycemia, and diabetes. What's interesting also about Oasis County is that cotton is our cash crop here. And then with cash crop, you can have agricultural practices that allow for pesticides that have arsenic in them to be applied because we don't eat cotton. So from there, if you're right side to side with a field that has arsenic applied to it, once you go through a cotton gin with it, you have all this waste afterwards. And most of the time you just end up burning it. So that leads to arsenic not only in the water, but in the air. So I think it'll be really interesting to see what happens when you combine both, the social and environmental aspect of it. I'm really hoping that it can shed light on any of the health issues out there, because a lot of the people who live out there tend to have cancer, they die a lot earlier, children get sicker, it's just a little more frequent. I'm hoping to show this with using R. I think all of us took Dr. Servo Boatwright's statistics and research class. I enjoyed it, and I think it'll be a really good way to demonstrate how both the answers from the survey and then what comes from the monitoring results will show. So hopefully the overall Texas project will help with not only the Nueces County colonias and just what's at the border, but any rural community in Texas. So, any questions?
You had one, and it deals with the survey aspect of the study, and I want to bring this from a fisheries side of things. Is this going to be a mail-out survey or an in-person survey?
In person.
Okay, and then the reason I bring that up is because the mail-out surveys, you get really, really low responses. But the in-person, because it is the colonias, you're gonna have a really good connection with the community to get good answers. Because a lot of times they see people like us as scientists, they just don't trust us. So you gotta have a really good connection with the community.
Thankfully, we do. So, yeah, so when finding the well locations out in the areas like there, we, Dr. Margulet has been in touch with, or for a really long time, a social scientist from Del Mar, who's also with the South Texas Colonial Initiative and For the Greater Good. So I go out with them. For the most part, it's been every week for about 3 weeks now to find people in those areas. So right now we have this down covered with well confirmations. And I would say, like, yes, kind of look at it as a scientist's point of view when you say, like, I'm from Maine, Corpus Christi, blah, 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 blah. But I think when you're with other people who look the part as well, it's a little more comforting. And also just, I don't know if age has anything to do with it or gender, just a more comforting factor, like being welcomed into someone's home and allowing for that to happen. So I haven't.
Good luck with your research.
Yeah, thank you.
That's a very good point, though. Absolutely, very important.
Thank you, Laura. And Amanda, don't let me forget, there's a comment in the chat that I'll share with you at the end. Our next presenter is Ahmed Gomaeid.
Hi everyone, I'm going to talk about, I'm Ahmed from Egypt. I'm working in Introductory, and I'm going to talk about the septic system failure and the water flow in the coastal regions. First of all, we are all reading for the previous literature. We encountered so many research gaps, like what are the factors that control the water table in the barrier islands, and what are the factors that control the interconnection between the surface water and groundwater, and the submarine groundwater discharge and the seawater intrusion. And is the pollution of the groundwater and surface water in the barrier islands and the coastal areas caused by the leaching from the septic system? According to all of that research gaps, we set our objectives, which is the prediction of the groundwater in elevation and the depth water in the barrier islands, and understanding the surface water and groundwater dynamics, assessing the impact of climate change, informing water resource management, and all of these objectives is aligned with the main goal, which enhancing understanding the hydrological process and enhancing the understanding the sustainable water resource management. The This figure shows the location map of our study area, which is located on the coast of Gulf of Mexico, and includes 3 counties, which is Galveston and Brazoria and Mitagorda. And these are our groundwater wells, which settled in that study area. Before I digging down into the research work, I want to emphasize some general outlines, like the septic systems. What are the septic systems? The septic systems are underground wastewater treatment structures. There are so many types of the septic system, and this figure shows one type of all of these types. And in that type, the air introduced into the septic chamber, and this air improves the survival of anaerobic bacteria, which is responsible for the treatment of sewage. And during all of that, maybe something like leakage or leaching out from this chamber. This leaching out, this sewage which leaching out may contain the fecal bacteria and nutrients, which pollutes and contaminates the groundwater in the study area. There are 2 cases when the leaching happens. The first case in high groundwater elevation, and the fecal bacteria and nutrients controlled by so many factors in that case, like the salinity of water, temperature of water. And the second case is low groundwater elevation. The fecal bacteria and nutrients leaching out into the soil, which controlled by its survival, controlled by the soil type and soil moisture and the organic matter in that soil type. Understanding the relation between the precipitation and the seawater level and the groundwater in the study area is crucial to know the flow path of this pollutants or contaminants. This flow chart shows the data and methods that we used in our study, which starting with collecting the data through 3 field trips per month. We collect all the water samples from all water bodies, like groundwater, like pool water, like seawater, and then we can make analysis of all of these analysis, nutrient analysis, oxygen isotopes, nitrogen isotopes, and we collect some field measurements like continuous data of groundwater elevation, depth to water, salinity, and we download a lot of data like tide, seawater level, and precipitation, and so many types of data. And we collect all of this data to make the pre-processing, filling the missing gaps, make normalization, make the transformation to get our results. This figure shows all the continuous data that we have got in the study area, which is tide and wind direction and wind speed and discharge in the stream gauges and our groundwater wells and some groundwater wells from the upstream. And this figure shows the GBM data that we collect for precipitation from the satellite or radar. And this figure shows the land use cover in the study area, which is the urban represent small amount of that land use cover, and most of the remaining of the study area is vegetation. This figure shows the relationship between the tide and groundwater elevation and precipitation in the study area, and there are something like collinearity and similarity between the precipitation and the heavy rainfall events and the high groundwater elevation, but that's not enough to make our results. This figure shows the discharge from stream gauge, one of the stream gauges in the study area. And this box plot shows the temporal resolution of the bacteria in our study area. And this shows a temporal resolution of nitrate, and this shows a temporal resolution of ammonium. And this figure shows the representative of bacteria and ammonium and nitrate in the study area, and we can see obvious inversion relationship between the fecal bacteria and ammonium in the study area, which impacts the importance role of this fecal bacteria in the nitrification process and convert the ammonium into nitrate. This figure shows that there are no linear relationship between the bacteria and the groundwater elevation, which proves the other factors that may affect on the bacteria in the groundwater, like salinity and temperature. This figure shows the the heat map of the study area. We collect all the parameters that we have in the study area and make a relationship, different person coefficient relationship between all the parameters, and we can see the inversion relationship between salinity and depth to water, and direct relationship between salinity and the groundwater elevation. We run some machine learning models to know all of this relationship to predict. We can use the past this past information and data to predict the future. We use all of this predictor to predict the groundwater elevation, and our R squared was 0.7, which is so pretty. This is the okay. Thank you. Thank you.
Any questions?
This is very preliminary. You should mention that because this type of analysis are going to be revisited with the proper transformations of data, and more investigations will be done. But this is just an initial assessment that you did, right?
Thank you, Ahmed. And our next presenter is Justin Elliott.
Hello, everyone. I want to talk a little bit about some of my PhD research. So I'm in Dr. Abdullah's lab, and the title of my talk is going to be the simultaneous quantitation and discovery of organic acids in complex mixtures. So to kind of get things started, I want to revisit the kind of omics revolution or the central dogma of biology, where we might have genomics studying DNA, what code is present, kind of what's possible in an organism or a system. Transcriptomics being what genes are expressed, proteomics being the translated RNA turned into strings of amino acids to make these proteins. And then you have these metabolites that are studied in metabolomics, which can be these small organic molecules that are either directly kind of affecting things upstream or are like the downstream products of the other omics that we see here. And so if we think about a system or an organism, we can think about metabolomics as kind of being the true phenotype or like the true state of an organism, where everything upstream is kind of like what's possible. And so metabolomics can be studied at various different levels. You can have a traditional approach where you're looking at an individual organism, or kind of zoom in a little bit and study individual macromolecule interactions. You can zoom it out and start looking at ecological interactions, seeing how different organisms might interact with each other. Or you can zoom out further and study entire ecosystems. Now, my research focus is going to be on the ecosystems or on the biosphere level. So trying to understand biogeochemical cycling of elements that are essential for life. But you can even zoom it out further and use it to study kind of planetary sciences as well. So the type of metabolites I'm looking at is often going to be referred to as dissolved organic matter. So these are going to be the small organic molecules that we find within the ocean or dissolved in any kind of water body. And you can think about it as like the phenotype of the ocean or the phenotype of the larger environment around it as well. And we want to study it because it has a lot of very important roles, both locally and globally. So you can use it to study a lot of anthropogenic effects, whether you're monitoring certain pollutants. But you can also use it to kind of zoom out and study cycling as a whole, especially for carbon. So there is a bunch of carbon that's stored in the ocean as dissolved organic matter. And so studying how that carbon kind of goes in and out of that pool is really important to understanding global climate processes. Additionally, we have lots of organic carbon that is really, really old, that has persisted for thousands of years. So trying to understand why certain molecules persist for these long periods of time versus kind of quickly turning back into CO2 is a major area of interest as well. Dissolved organic matter in any kind of water system is a highly complex mixture. So you can have tens of thousands to even millions of different organic molecules in a single liter of water. So kind of coming up with some sort of analytical technique to be able to characterize all of that is really complicated. And so overall, there's been a lot of instrument biases that have led to some major blind spots in our understanding of dissolved organic matter. So how do researchers go about trying to understand what's within a complex mixture or what metabolites you have in your sample? So you usually have this one approach where you're combining two kind of sophisticated techniques, where you might have a separation technique, most notably in the form of high performance liquid chromatography. This is going to be the most common approach, where you're separating compounds into more manageable batches. So instead of trying to understand all 10,000 molecules at the exact same time, you can kind of take it in small bite-sized pieces. And then once you separate it, you can then introduce it to your mass spectrometer, where you can have these molecules ionized. Once they're ionized, you can guide it and move it in such a reproducible way that you can kind of try to identify your molecule by getting the mass of your entire molecule, breaking it down, and then generating like a molecular fingerprint to understand what exactly you're looking at. So I quickly want to turn our attention to high performance liquid chromatography, or HPLC. And so this is the most common approach, and it is very well suited to separating things that are nonpolar and positively ionizable, because it separates analytes based off of their hydrophobic interactions. And so although it is a very well-studied and easy to access, really commercially available, it doesn't separate out organic acids that well from each other. And so you can separate out a wide variety of different analytes, but you do get this kind of batch of analytes, most namely organic acids, that kind of fall out of your analytical capabilities. So ion chromatography is a unique variation of liquid chromatography, where it is designed for separating out these highly polar or these ionic molecules. And so ion chromatography separates out ions based off of their ionic interactions. And so it becomes really good for separating out organic acids from each other. And it's been around for a while, but only recently became really compatible with mass spectrometry. So this has been where we decided to turn our research interest to try to understand the negatively ionizable metabolites that makes us a little bit different than a lot of other research groups. Now, you might be wondering, why do I care so much about looking at negatively ionizable metabolites? Oftentimes, the very central metabolic pathways for all organisms are going to involve organic acids. So here I have a large kind of metabolic metro map showing these highly conserved metabolic pathways in various walks of life. And if we think about the major components that are involved with building life or maintaining it, they're all kind of involved with organic acids. So you can think about amino acids, the formation of proteins, fatty acids to make your cell walls, your citric acids, and carboxylic acids for energy production. And so if we want to understand life or how life interacts with the environment, we need to be able to understand organic acids and kind of how they're changing. There is the other side of the metabolomics approach. So there's the mass spectrometry side, and mass spectrometry has traditionally been either targeted or untargeted. So targeted is like where you're trying to maximize your instrument sensitivity and your selectivity to try to achieve quantification. So you can give you some sort of numerical value as to how much of your analyte is there for your specific target. Now, if you kind of turn the dial all the way to the opposite end, you then have an untargeted type of analysis, where you're trying to maximize your spectral coverage and understand what's in your mixture. And so this is used to kind of generate new hypotheses or find important biomarkers in whatever sample you're looking at. So what we're trying to do is try to combine those with this new technique called SQUAD. So this stands for simultaneous quantitation and discovery. And this can be done with the kind of new modern mass spectrometers, which have multiple mass analyzers working at the same time. So you can kind of be conducting multiple experiments with a single injection. This is really important because a lot of times you have samples that aren't replaceable. So we hope to kind of combine these two approaches by just essentially running calibration curves and using unknown libraries to conduct both quantification and discovery type experiments. So overall, the goals of my research is to create this massive organic spectral database and be able to implement that to run the first kind of SQUAD analyses using ion chromatography mass spectrometry. Once I have this method down, I want to then use these tools to study dissolved organic matter transformations across various water masses and hopefully bring this tool, make it available for other people to do more traditional metabolomic studies as well. Thank you.
Time for one quick question for Justin.
Yeah, I've got a question. This is Evan again from the Water Board. So first, I have to say that's probably the most complicated flowchart I've ever seen in a presentation before. That's saying a lot because I've made some complicated flowcharts. So I think this project would be really interesting in combined with the acidification project because those are kind of related. And the comment I had is we really struggle here. In fact, we just had a thing like maybe 2 or 3 days ago where we were trying to say, well, what can we collect as part of our sampling? And, organics is always one, and dissolved organics. The problem is that we don't have a mass spec and don't really have access to one. So if through your studies you could find comparisons to cheaper systems of taking data or ways in which us governmental agencies that take data could be taking this kind of data on the cheap, those would be really great things for us to know.
I can't think of any suggestions off the top of my head right now for kind of making this cheap. Kind of what makes it possible to look at organic matter and understand it very carefully, especially the discovery type of stuff, is going to be access to those fancy mass spectrometers. However, if you do kind of are able to come up with a handful of target kind of important biomarkers and stuff, then that would be pretty easy to then send that off to some sort of lab that does have a mass spectrometer and run a kind of routine analyses to look for those biomarkers. But that's about the best I got for you. Thank you.
Thank you, Justin. Our next presenter is Allison Juras.
Thanks. Yeah. Just buy me one. We can do.
Just have a USB thing.
Next, right side of the screen.
Just a click.
They can be monthly.
Okay, good afternoon, everyone. My name is Allison Gerard. I'm a PhD student here in the CMSS program, working with Dr. Felix and Dr. Abdullah. And today I'll be talking about the use of Orbitrap mass spectrometry for stable isotope studies. So first, my clicker's not working. Second, a little background on stable isotope biogeochemistry and a reminder of what isotopes are. So isotopes of an element will differ only in the number of neutrons in their nuclei. So because they're the same element, they have the same electron configuration, same chemical behavior, but they will differ in their physical behavior. So essentially any physical, chemical, or biological process is going to cause something called isotope fractionation, where that process prefers to use one isotope over another, just based on reaction kinetics and energetic favorability. And what this means is that different materials can be characterized by what we would call isotope signatures that sort of reflect the various processes that material has undergone.
So this has been exploited in a wide variety of research fields since the 1950s, from all sorts of things like pollution tracing to food web elucidation, forensics, archaeology, all sorts of different research fields have implemented stable isotope studies. So some of the theory behind this technique called isotope ratio mass spectrometry is that it's this sort of relative study where you measure the ratio of a rare to abundant isotope in some material, and then you compare that ratio of isotopes to the same ratio in a standardized material. And it sort of allows you to say that some material is enriched or depleted in a rare isotope, and then you can start to sort of compare different sources of some material and draw some inferences about the processes that that material has undergone. So I have an example here. It's not my data, but from this paper from 2013, looking at nitrate in surface waters. And we can use these isotope signatures or delta values to get an idea of where that nitrate came from. So you can see on the x-axis, they have the isotope signature for nitrogen in the nitrate. And then on the y-axis, they have the oxygen isotope signature. And these different boxes sort of plot these different sources of nitrate. So you might be able to say that your nitrate was sourced from fertilizer, or it came from the atmosphere, or it came from the ocean. All of those different sources of nitrate have different isotopic signatures. So the instrumentation that has generally been used for these techniques uses these magnetic sector mass analyzers, which will separate molecules based on their different mass-dependent trajectories through a magnetic field. So you can see that magnet there. This is going to be your magnetic analyzer, and you're going to send a beam of ions through that magnetic field. And then based on their different masses, they're going to sort of separate into their own ion beams. And then there will be some detectors at the end of that waiting to collect those signals. So it's a really powerful technique, but unfortunately there's only a small selection of molecules that are amenable to this technique because you have these really precise detectors set up at the end. So this is sort of inherently problematic. If you're trying to study carbon or oxygen or nitrogen, you're going to have to convert all of your compounds of interest into these molecules that can be studied with this technique. And you're not always trying to directly study CO2 or N2. You might be trying to study these larger molecules that contain those elements. And it can be done, but there's also some other limitations of these techniques. The sample preparation is pretty extensive. And when you make these conversions so that you can get these analytes of interest, you're losing some information about the chemicals that you're studying. So you can lose some compound-specific information. You don't exactly know where those isotope signatures are coming from. And it can be costly. There's a lot of consumables involved. And some isotope systems simply can't be studied with this technique. So now I will introduce to you Orbitrap mass spectrometry, which has generally been used in life sciences and has become incredibly popular in a lot of those research fields because it's really, really powerful in terms of mass resolution and mass accuracy. And it operates on a totally different premise than our magnetic sector-based instruments. So this Orbitrap mass analyzer is this little football, spindle-shaped contraption here, and it's a Fourier transform technique. So what ends up happening is your ions sort of oscillate within that little spindle and generate a frequency signal that can be transformed, and it imparts this really high mass resolution. So that's been really important, as I noted, in some of these life sciences field, particularly these omics studies. So things like genomics, transcriptomics, proteomics, where you're studying these really large molecules and complex mixtures. But some of the properties that make this technique so useful in those fields will sort of, have raised the interest in terms of using this for isotope studies. So when we consider some of these features and question, how does this offer improvements to our traditional isotope ratio mass spectrometry? Well, what we find with Orbitrap is that these instruments have this ability to resolve these really small differences in mass, and not just a single atomic mass unit, but fractions of an atomic mass unit. So now instead of just saying, I have a substitution with a 15N isotope versus the more common 14N isotope, you can actually differentiate whether you're substituting that with 15N versus a 13C atom and so on and so forth. And this offers this opportunity to unlock these new isotope signatures. Okay, so sometimes we have compounds that are substituted with several rare isotopes, and those molecules become really, really rare, but Orbitrap has the power to sort of unlock these new signatures. And you can see some of the different isotopologues, as we would call them, for nitrate, where you have all sorts of different combinations of nitrogen and oxygen isotopes. And in terms of sample preparation, throughput, Orbitrap is going to offer all sorts of improvements across the line. So, ideally we would be able to just hop in and use this, but there's some considerations that we have to keep in mind during our method development. So we have to avoid this isotope fractionation in our preparation of our samples, right? If we're trying to study the isotope ratio within a material, we want to conserve that natural ratio. So we need to make sure that our instrumentation and our method isn't imparting any fractionation along the way. Of course, we want our results to be reproducible throughout different fields, throughout different labs, and not just reproducible. We want to make sure that we actually are seeing significant improvements to the existing methods, right? The conventional methods have been accepted for a long time. So for this to hit the mainstream, we want to offer significant improvements with this instrumentation. Now, for my research, once we get through the sort of method development phase, of course, we're going to have some applications within the coastal and marine systems fields. So we have some potential applications sort of picked out right now, probably something to do with nitrate source apportionment in the Texas coastal waters, nitrate being a sort of marker for pollution and anthropogenic effects. And then we might take that to a different sort of environment than we're used to here, potentially looking at nitrogen deposition in places like alpine ecosystems. But really the possibilities are endless for these techniques. If we can actually get through the method development phase, then you can essentially see this applied anywhere where the traditional techniques have been used. So we're really excited to have this project underway and hope that we can reach out to other groups and see how they might be able to use these techniques. And with that, I will thank you for listening and answer any questions that you might have.
Any questions for Allison? We have time for one. Yes.
The fractionation process has so many equations and so many equations to calculate this. What will you do and add for this?
So, we can basically study the fractionation based on, there are some sort of theoretical, markers that say, oh, we expect the fractionation to be this extent for this process, or something that, like an example would be something like industrial. Okay, if you have like the combustion of fossil fuels, usually there's not much of a preference there if it's going to use 13C or 12C or 14C because you're just combusting everything, right? So you expect minimal fractionation versus something that's maybe a slower process. You're going to see a more dramatic fractionation. So there's some sort of like theoretical stuff that goes into that. For me, it's not necessarily identifying the fractionation factors, but sort of identifying those signatures using the analyses and then sort of matching those to what is known or what we can study elsewhere.
Thank you, Allison.
I'm going to make a comment quickly. You can tell you have been teaching.
Thanks. Yeah,
It's a good experience. Everybody remember this. Teaching, it's a great experience that really improves your presentation skills. You can see that.
18, 19-year-olds. Our next presenter is Remy Lavelle.
Hello, everyone. My name is Remy Abe. I work in Dr. Crocker Siemens lab, same lab that Dee works in. I'm part of the Masters in Chemistry program here at TAMUCC. And my research doesn't have to do with PFOS. It has to do with benzoapyrene, which is a molecule I'm going to hopefully not scare you too much about because it is in the environment, and what we study is daily environmental exposures. But what I'll be focusing on in today's talk is the in silico assessment of how this molecule may be affecting or inhibiting epigenetic processes in living organisms. So a little bit of an overview. I'll kind of introduce benzoapyrene and how it is affecting human or non-human organisms, and how could that be affecting us human organisms. And then looking at the in literature hypotheses as to how this can be happening in the first place from a chemistry point of view, not from a biology point of view. And then I'll be presenting my own kind of novel hypotheses as to why or how this can happen in the first place using kind of organic chemistry logic, and then testing that logic through in silico assessments. Right, so this molecule right here is our antagonist in our story. It's benzoapyrene. It's a polycyclic aromatic hydrocarbon that we are exposed to on a daily basis. But before I kind of delve into this molecule, I want to ask you some questions. So first of all, have you ever smoked? Have your grandparents ever smoked? Have your grand-grandparents ever smoked? Okay. Next, barbecue. Has anybody gone to barbecues? Yes, of course, we're in Texas, right? What about Corpus Christi? Has anybody lived in Corpus Christi? Has anybody worked next to or close to Corpus? Yeah, I do. So these are all sources of benzoapyrene. Usually it's produced through a combustion reaction which didn't complete fully, right? So this carbon is going to form into polycyclic aromatic hydrocarbons like benzoapyrene. Right, so then once this molecule is in the environment, then we can be exposed to it. And what happens to what happens to organisms when that happens? So here we have a model organism called Medaka. It's from a model organism from Japan, and it is used to study human diseases. It's a great model organism because development of this organism is very similar to our development, and also it's very cheap to raise compared to rats, and it's transparent. So then you can see in the organism what's going on. Now, in an exposure experiment to benzoapyrene, what we see is that in the first generation that is exposed to benzoapyrene, we have a phenotype arising, actually multiple phenotypes arising. So osteoporosis, neurotoxicity, and many others. Right? And then in our control group, these are healthy Medaka organisms, right? And then if we take these organisms and then we look at the next generation, but make sure that they're not in an exposed environment, we still see the same phenotype arising. And it's consistent in the F1, F2, and F3 generation. And that's why I was asking the question about your grandparents and your grand-grandparents and your parents as well. So this is quite concerning if this is to be also translated into humans as well, right? So exactly how is this happening? What we do know for sure is that benzoapyrene is metabolized by the organism into a multitude of different compounds. But the main compound of interest is benzoapyrene epoxydiol. It's this molecule right here. And as you can see, it's has some electronegative side and electropositive sites, right? But the main thing that I want you to notice is this oxygen group right here. That's an epoxide. It's extremely reactive compared to other oxygen functional groups, and is mostly and most likely the reason why this molecule is so toxic. But I want you to think about this past figure, why you believe then this phenotype or multiple phenotypes are being inherited consistently. Most of us have gone to school and probably know about DNA and think that, oh, it's a mutation. It's inducing, benzoapyrene is inducing some sort of mutation and thus leading to toxic effects. But actually that is not the case because the phenotype is consistent. So we see them consistently throughout the generations. And if it was a mutation, we would expect that phenotype to be random, not consistent. So we have to reject that hypothesis. Now, what could be the reason why this is being passed on? It has to do with another layer of information called epigenetics. And I'm not going to talk about that today because it gets a little bit complicated. But essentially, we have many different cells, and how do these cells know what to do and how to look differently, how to behave differently? It essentially has to do with how genes are managed, right? And so we have proteins and many different codes that allow us to regulate genes specifically based on what kind of cell type is present. And so one example of how this could be happening, or one proposed mechanism of how benzoyl is leading to inhibition or leading to dysregulation of the epigenome, is by, yes, binding into the DNA, forming DNA adducts, which is modifying one part of the epigenetic called DNA methylation. But I have, there's some issues with this hypothesis. First of all, this is the explanation for why this molecule can be carcinogenic at high elevated concentrations, but it doesn't explain what we see in our experimental observations. What I do want to propose very simply is that the enzymes and the proteins that are organizing the DNA are actually also interacting with benzoyl. And because benzoyl is an a nucleophilic molecule, then it can do the same chemistry that other molecules that we have within our body do as well. So preliminary studies that actually I did during my undergraduate found docking sites in some of these enzymes that do these reactions. And what I would like to show is, or test through molecular dynamics, is that, sorry. Oh, wait, how do you make that there? Okay, through molecular dynamics, is that this molecule, benzoic pyrene epoxydiol, is capable of reacting with enzymes and within the biomolecules of the epigenome. And this is an example of how these simulations allow you to predict whether or not this is possible in the first place, because analyzing that through mass spectrometry or any other techniques gets very, very complicated in the first place. So thank you very much.
We have time for one question.
One of your slides, if you go back a little bit, which one? Does it have anything to do with macaroons?
Which one?
Go back. You probably want to exit. The one that has, I can't remember what it was about exactly, but I was just trying to make a joke here. It's not, you're not finding it. They look like macaroons. Yes.
Yeah, it looks like it.
I thought you know,
Oh, yeah,
and macaroons. That's just.
It does look a little bit, right?
Oh, you just want, I'm going to buy some now. Do they? Sorry, there was a question, a real question, not what I had.
When you're breeding them from first to second generation, are you breeding them with two exposed ones or exposed, non-exposed?
So the parental generation is exposed and the following are not.
Okay.
Yeah.
Okay.
Our next, thank you, Remy. And then our next presenter is Quincy Walker.
All right, so my name is Quincy and I'm a master's student in the CMSS program. My background is actually meteorology, so it's a bit different than what we've been hearing today. So I'll be talking about two research projects that I've been working on, both ongoing, and the common thing between both is that I'm using radar data to analyze isolated extreme weather events. gonna skip this slide. So the first research project I'll be talking about is the characteristics of radar-defined tornadic convective features. And essentially what we did was that we pulled the tornado reports between 2017 and 2021 and matched them to their parent storm. And then the database I was using was Dr. Liu's convective feature database, and it reads in composite radar reflectivity data, and then an ellipse is drawn around these clusters of 40 DBZs, and 40 DBZs associated with extreme rainfall or ice and hail. And so once these clusters are found, then an ellipse is drawn around it, and then we then temporally and spatially co-located the two events together. And so yeah, I'll talk about some of the data that I got from that. So one feature that we took a look at was the actual size of the convective feature itself, so that area within that ellipse. And we found that despite producing the strongest tornadoes within the data set being the EF4 between this time frame, their convective feature size is actually the smallest significantly when comparing to EF0s or EF1s in their parent systems. Then we looked at the system morphology. So just going back to that ellipse I was talking about earlier, we took the ratio between the minor axis of the ellipse and the major axis. And then with that ratio, of course, you get a value between 0 to 1. And so values closer to 1 are associated with smaller or with rounder systems rather. And you can associate these with supercells, so these isolated events right here, and then values closer to 0 are associated with squall lines, so these very long chains of storms that can span across several states in some cases. And then we found that the EF4s, the systems responsible for producing an EF4 had the roundest systems within the data set, as these are likely associated with supercells, and then the EF0s, EF1s are associated with the longer squall line systems. And this is kind of why, as I said previously in the last slide, that this convective feature size of the systems responsible for EF4s are significantly smaller than EF1s, just because the EF1 systems, they span across several states in some cases. Then we looked at the orientation of the convective features in relation to their propagation angle for the tornadoes themselves. And so as you saw in the previous slide, a lot of them were kind of pointed towards the northeast direction. And we found that the tornadoes have a propensity to propagate in the same direction that the CF is oriented. So if it's pointed towards the northeast, then it's likely the tornado is likely gonna travel towards the northeast. Then currently as a master's student, once again, I'm using radar data, but I'm analyzing the properties of extreme rainfall events using the GPM precipitation radar. And so this data set spans from 2014 through 2022. And as mentioned, or similar to the convective feature database, as I mentioned for the tornadoes, the extreme precipitation feature database reads in the data the same way, but from the GPM satellite. But in this case, it clusters up or groups together clusters of pixels with a near surface rain rate of 20 millimeters an hour. And then between this time frame, I have 360,000 events within this analysis. So this is like the centerpiece of the research is the geographic distribution of the EPFs based on their near surface rain rate. And so I broke them into several categories, starting from 20 millimeters an hour all the way up to exceeding 200 millimeters an hour. And so we found that in general, most of these EPFs occur over the ocean, but over land, there's some pretty noticeable hotspots, as you would expect in the Amazon, Central Africa, South Asia, even in the Gulf of Mexico here. And so one thing that we are going to take a look at is how many of these are results of hurricanes or tropical cyclones. And so moving on to the next slide. So we have, we compared the relationship between the near surface rain rate and the DBZ values of the EPF itself. And the main takeaway from this is that the color fill in the background is the ocean EPFs, and then the contour lines is the land EPFs. Is that you can have a similar near surface rain rate, but over land, the DBZ is actually a lot higher. than over the ocean just due to the particle sizes between a land EPF and an ocean EPF. And then we also looked at the heights of the storms. So I plotted the longitude of the EPFs compared against the rain rate as well as the height of the storm. And generally, you would probably expect that it's generally accepted that the taller storm is likely stronger just due to convection and the strength of the updraft. And you would probably think that these might have the highest rain rates within in nature, but it actually turns out that may not be the case. So in Africa, for example, within the data set, their storms are actually the tallest within the data, but compared to Asia, their systems are actually the shallowest, but it actually turned out that the rain rate for the Asian EPFs are actually a lot higher on average than the African EPFs. So yeah, there's this kind of a connection to a study that was done a while ago. I think they were using a TRIM satellite to do this analysis, but this same trend also appears within my data set. And then, but yeah, that's it.
Thank you, Quincy. Any questions for Quincy?
Yes. Let's be here.
And our next presenter is Mahima Yogesh.
Okay, hello everyone. As Dr. Douglas mentioned, my name is Mahima. I'm really excited to be here and tell you a little bit about the research I've been doing. So a little bit about me. This was something that I shared in the first meeting that we ever had, but I received my Bachelor of Science in Environmental Science and minor in Oceanography from Texas A&M University in College Station. And now I'm a second semester, first year Master of Science MARB student here. And I'm conducting a lot of microbial ecology work under Dr. Yadwan Lin. Some of my research interests are marine microbiology and ecology, and isotope characterization and ecology. That's stuff that I'm actively working on right now. Some of my other interests that I hope to delve into at a certain later time are coral reef microbial ecology areas of study, as well as climate policy and justice issues. So yeah. So my project is actually a year-long time series study conducted in the 2 sample sites in the Gulf of Mexico, one being kind of coastal adjacent, and the second one being open ocean. And my project started technically in October of last year, and so we have been going out about every week collecting seawater samples, tar balls, like the little ball that you see on the beach, and sargassum samples that get bagged. And so that's been something that's been going on since last year. And the goal of this project, there's 2 objectives. The first one being to characterize the water column microbiome in 2 areas within the Gulf of Mexico, coastal and open ocean. I'll have a picture like in the next slide to show you guys where my sampling sites are. And to achieve objective 1, we're hoping to sample seawater, the sargassum and tarball samples, and then down the line, ideally this semester, the plan is to conduct DNA extraction on those seawater samples, and then hopefully that'll give us a better idea of the question of who is living in the water. And yeah, and then the second objective is to explore the dynamics of the isotrophs in coastal water environments, including the water column. And so again, that DNA extraction that we'll be doing will also be able to achieve the second objective. We'll use a specific marker during the polymerase chain reaction to see if there are isotrophs in the water. And then down the line, we're hoping to conduct something called the ARACOS method. It stands for, and it's a mouthful, but it's the flow through incubation acetylene reduction assay by cavity ring down laser absorption spectroscopy. I had to write it down, but that will basically allow us to quantify nitrogen fixation rates to see if different diazotrophs are conducting different levels of fixation. So I forgot to mention this, but the reason that we're doing this is because diazotrophs tend to be understudied, especially in a region like the Gulf of Mexico, and they're really crucial. They help make a lot of nutrients bioavailable within our oceans. Recently, there's been a study put out about how there's a great sargassum belt that's kind of just been like getting found in like the Caribbean, and it's just like a huge belt of sargassum, and they found signs of diazotrophs growing on there, which is amazing because the paper found that a lot of these diazotrophs could potentially be providing this sargassum with essential nutrients, which I think is really interesting. On a climate change basis, I think that everything being interconnected is super relevant. I think that if we see changing ecologic or changing environmental conditions, we could see changing rates of diazotrophs present in the water column. And the same thing goes for my first objective, which is to look at cyanobacteria. As you guys may or may not know, cyanobacteria form the base of the marine food web. They also help with nutrient cycling. They make oxygen available. They some of them can also be diazotrophs, so there is a bit of an overlap. So this also just goes to say that this work really is important, and it's also relevant that we're in an El Nino period right now. So looking at the warming effect on the ecology of the microbes is super crucial. And I'm not there yet, but it could definitely be interesting to see what is found in terms of who is present in the water column and what role they're playing and the different ratios of each type of organism. So though I'm not there yet, I'm really excited and nervous to see what that might yield. So something that's nice about my project is that I was able to get some DNA water filter samples from my professor, Dr. Lin, and Dr. Xinping Hu. They went on a research cruise in the Gulf of Mexico, and their samples, there's about 13 of them. It's the left photo. And as you can tell, they have a couple of coastal water samples, but a majority of them are in the open ocean, and that provides my project with spatial variability. And my two sampling sites are the Packery Channel, which is the lower of the 2 points, and then the Horace Caldwell Pier, that's in Port Aransas. And so the blue side is the Gulf of Mexico, so both of my sampling sites are facing the Gulf of Mexico, so that's kind of what's going on right now. And kind of as a basic project workflow, I did kind of touch on this, but right now we're still in the sampling phase of things. So that's the sampling of the seawater, sargassum, and tar balls. This semester, the goal is to conduct DNA extractions on all of my filtered samples, as well as all of the samples from the cruise. That's about 40 sampling samples across 13 sampling sites. Hopefully during the summer in semester 3, I can utilize the data from the extractions and PCR to determine, again, the question of who is in the water column, what is the ratio of organisms within the water column, how is that changing? And then the fourth objective is, or the fourth part of the workflow is to conduct that ARACOS method to quantify the nitrogen fixation rates. Hopefully that'll be, again, semester 3. And then, last part, putting together all that information to further understand how are the ratios of organisms, microorganisms changing in the water column? What does that mean? Can any of these organisms be used as a proxy to predict changing environmental conditions? One of the papers that I used for a literature review found that different ratios of cyanobacteria could be used as proxies. There are different ecotypes could be used to predict changing water conditions. And I think that could be really interesting. And yeah, that is the goal as it stands right now. Just some photos from sampling. It's really beautiful out there. This is from the Packery Channel. So we toss a bottle into the water and pull it out, and we get right on those rocks. And this is our new sampling site on the pier. And yeah, it's really exciting to be out there. I'm very thankful to be able to do this. And I appreciate the opportunity to talk about my research through stages. And yeah, I'm open to any questions. Thank you. Yes.
I saw in that last picture for the sampling, have you guys thought about maybe getting shoes with spikes on them?
So I did invest in a pair of water shoes because for yeah, that one, the algae on the water is definitely a cause for concern.
The barnacles will slice your feet.
Right. I know I'm barefoot in that photo. I actually presented this to some 5th and 6th graders. We collaborate with a teacher, an amazing teacher at Flower Bluff Intermediate, and they made fun of me. They were like, "Why do you have water shoes on?"
So I did invest in a pair, but it hasn't solved all my issues, which is in part the reason that we switched to the second site. As you can see, it's like right on the water, so there's no requirement to like potentially get your feet ripped out, which was a big incentive for me, so. It's, yes.
Big picture question, and this has to do with the sargassum
Mhm.
and your objectives. In the last 2 decades, there's been a dramatic shift in
Mhm.
the entire western side of the Gulf in the distribution, spatial amounts, and the abundance of sargassum.
Mhm.
We don't get it like we used to at all. And we haven't had a good sargassum wave in probably 10 years.
Okay.
So how does that affect your research?
Right now, ooh, that's a good question. We've noticed like the seasonal pattern of there being like more in the summer and less in the winter, but like you said that there was like less of, like of late, there's been like less of the stuff.
I remember going to the beach as a kid and it would be stacked up at the front of the beach to
Right.
your waist.
That's, yeah, that definitely changes things. As far as I'm concerned, like the belt is like kind of free flowing, and I guess that something that I would like to see is like the extent of it, especially during the summer on the beach. I don't know if that completely answers your question, but big picture wise, I'm hoping that if I can at least like understand the ecology of the microbes on the small sample size of sargassum we can understand maybe how that nutrient availability influences their ability to grow and propagate. But it is interesting to see how, I mean, I've never seen it up to my waist, so that is crazy.
It's orders of magnitude fraction of what we used to get on the beach here every spring.
I definitely, that is significant. I did not know that, so that'll be interesting.
As a fisheries biologist, we think that has to do with the decline in greater amberjack and gray triggerfish because they rely on that for the juveniles. So we don't get them this side of the Gulf anymore.
Interesting, yeah.
Sargassum is a big thing.
Right.
The Caribbean still has it, right?
Oh, yeah. That's where it
I've
all
seen
goes
it
now.
in 2018, there were piles of
Yeah,
it.
it all goes to the Caribbean and
Yeah.
the eastern side of Florida.
It's actually becoming a problem for them. Maybe you can get some samples from there if you want to study.
I would love to be able to go to the Caribbean and get some sarcastic samples.
Okay, thank you, Mahima. And we have one last presenter today, Ivani, you're up.
Hello everyone, my name is Frank. I will be speaking on assessing the impacts of extreme wet events on groundwater pollution in unincorporated coastal communities in southeastern Texas. Okay, going forward, this is the outline of my presentation. I'll run from introduction down to the references. Okay, so climate change has really impacted the globe in a very quick way. We've experienced intense wet events, intense vegetation, and with the high rainfall events, we've noticed there is progression of contaminants into the groundwater system, which affects a lot of people. So going forward, our motivation for this study is to be able to understand the dynamics of the groundwater contamination in some coastal areas. So our study area is actually the Aransas and Nueces County. So looking at this map, you would understand that the Aransas and Nueces, they have high flooding from this graph here. This is the Aransas County, and over here is the Nueces County. Sorry, mouse. Okay, this is the Nueces County. So these regions have been known to have high flooding, as indicated from the map of Texas flood used by Angela. And then the communities of interest are the unincorporated communities. So these communities, they do not have a formal body of a formal body or organization that would actually speak for them or try to solve their environmental challenges. And with that, it makes them more vulnerable to a lot of contamination issues. So when they have these kind of problems, they tend to solve their issues on their own. So recent surveys, we discovered that there are issues of cancer, issues of stroke, and a lot of other health issues. And the big question is, what would be the cause of these health issues? Could it be that they are that the water is contaminated from a lot of, contaminants from maybe landfill leachates, industrial effluents, and the rest of them. So I'm going forward to my research objectives. So the first one is to estimate the groundwater recharge in response to direct and indirect effects of climate and socioeconomic inequalities in the area. So with that, we would try to carry out a high resolution calculation of the critical and saturated water zones using our geoprobe. So with the geoprobe, we intend to one, understand what happens in the subsurface. So with the HPT, we'll understand the formation permeability, and with this NMR, we'll understand the volume and the volume of the groundwater, and also for the CBT, we'll understand the subsurface stratigraphy, which will help us to understand how contaminants move in the subsurface. All right, going forward, so this is just a picture of what's a log looks like the HPT log. So what we intend to do is to correlate these three logs when we've gone out for our field work, and then be able to understand what happens in the subsurface. So going forward, second thing is to carry out determine the groundwater contaminant type sources and followed in relation to the community and environmental and socio-economic attributes. So we collect 30. We'll collect samples from 30 wells and 10 surface water locations monthly over the period of 3 years. And with that, we'll carry out our lab analysis in my advisor's lab, the Muglets, and then carry out temperature changes related to inputs of the contaminants. Then going forward, also predicts the groundwater quality as a function of hydroclimatic conditions. So here, we just pick out the parameters of the PO, pH, and ORP, and then compute entropy weights based on quality models. Also, we intend to build some machine learning models by using groundwater elevation, temperature, and pH to predict the different kind of contaminants we have in subsurface, like the lead and the nitrates and whatnot. So the last thing I'll be working on will be to identify the possible PFAS compounds in the groundwater of these communities. So with a sample, as usual, take samples from these areas and then take it to the lab. We will be shipping out our samples to Eurofins for testing. And then when we get a result back, we'll conduct hierarchical plus analysis to understand this matter of the PFAS, and then carry out some manual forest application as well. So in conclusion, we aim to have a deep understanding of how these high wet events influence the dynamics of groundwater pollution in these communities. And I personally would like to establish the fact that there is a need for a multi-level action to stem the prevalence of these events or these activities that tend to be a threat to the groundwater system. So I would like to thank my grant advisor, Dr. Mokulet, for drafting me to the ISS project funded by the NSF, and also to thank the Center for Global Studies for providing the grants for my PFAS analysis. Here's my references. Thank you so much.
Any questions for Ifani?
How is the NMR data being used? Like what NMR data are you collecting?
Okay, so we have the geoprobe, right? So this geoprobe would insert the log inside the subsurface, which would detect a lot of parameters for us. And then with that, we'll now calibrate and know what is happening in the subsurface. So mostly the NMR is just to determine the volume and pressure of the groundwater in subsurface, and also to help us quantify the aquifer porosity and permeability.
So what kind of samples are you collecting for the NMR data?
We are not collecting samples per se, but we are just, it's actually a magnetic equipment that sends signals and gets data in subsurface.
Oh,
yeah.
It's measuring continuously, it's pushing down to the subsurface.
Oh, so not like okay, different.
But you can.
It's not like you're boring your hole, like drilling downward, like borehole,
Okay,
and then with that, get data, get signals.
okay,
So
okay.
you're getting something like what he's showing there
Uh-huh.
for electrical conductivity, porosity. But it's a depth profile.
Okay.
And it's measuring every 2 centimeters, what that changes.
Included in the proposal. How can
The
you pick
NMR?
the location for the new well that you planning to?
Okay, we're considering looking at areas that are close to sewage dump sites, natural influence, so that we can help to get have a close proximity to these areas to be able to get the real data that we need.
You will not depend on the runoff or something like this, upstream and
We are
downstream.
considering every factor
Yeah.
to get this data that we need. So
Which type of isotopes you will depend on to determine the recent charge and older charge for this organic pollution?
well, I've not really considered that right now because I'm still working on trying to get these concepts together as a whole. So I think I will consider that when I get back to.
We'll be using oxygen and hydrogen isotopes as proxies for recent recharge to the aquifer, right? And we'll be using the radon and radium isotopes, which are also telling us about how long the groundwater has been in contact with the formation versus recent diluted by the precipitation, which is not, doesn't have this radium isotopes. But yeah, Ifan is just throwing out a huge project that he's trying to put his objectives together now, just giving us a pretty big picture right now of what he will dive into.
I think the tritium analysis for this
Radium?
yeah, for this type of projects is crucial because it the best way.
I know it's dating the groundwater, but we can use our own, not like quantitative dating tools like radium. It doesn't tell you exactly what the age of the groundwater is, but it's telling you if it's newer or it's older. Yes, qualitatively. So we could even, I mean, he will be able to determine recharge rates and do based on the chemistry determine those changes, but tritium is pretty expensive and very, very tedious to sample. And as you could see, it's going to be about 1,400 samples. So that is a lot for us to do tritium.
Thank you.
Thank you, Connie. Thank you all for your presentation today, letting us know what your thesis and dissertation research is going to be focusing on. And I'd like to thank all of our stakeholders and community partners that were able to join us in person and online. Hopefully this kind of is an introduction to our trainees and their interests and backgrounds. And we're looking forward to meeting with you guys and talking with you throughout the rest of the term.
I think you had a question in the chat that you wanted to ask for.
So there was a comment for Amanda earlier. Folks from IOB, from Ingleside on the Bay Coastal Watch Association would also be interested in the seagrass alkalinity talk and would like to see Corpus Christi Bay be part of the study, given the recent approvals for massive desalination plants in the Bay. And then for Laura, Evan had a comment for you. Many wells across the state are monitored by Texas Water Development Board and a good relationship with the owners, and they have a good relationship with the owners of those wells. So Evan's willing to be in touch if you want to contact him or if you want him to put you in contact with the groundwater staff at the board.
I would like to leave you all with an exercise, a homework for next time. Based on what you've seen today here and everyone's research focus, think about how what one, two, or three or more are doing is applicable to your research. How can you benefit from their knowledge to better inform your own research? Because this is what we're here for, right? To try to build this interdisciplinary skill. So I'm already seeing I could, connect and do that network, the neural network kind of, and lines drawn between all of you to where you could all benefit from that. So I would just take, like Ifani's and Laura's project, for instance, they're going to use precipitation, and intensity of precipitation is extremely important if you want to look at where flooding is going to happen. They will be, they're looking at toxicity, and with Dr. Fraka, she will look at expose her fish, those pretty fish, to polluted groundwater to see what the potential, the expected health impacts are for those communities as we are doing that social environmental study. Dissolve organic matter, and if you take that and the sources of pollution, right? Like we're talking about the fingerprinting. For seagrasses, think about how precipitation trends and patterns will impact those, as you already mentioned, doctors who study. But are we seeing more changes in precipitation patterns and extremes, like more downpours, how those are likely to impact differently acidification than just slow rain that's trickling in and infiltrating to the ground and so on. So just a few examples. Let's just think about who can you talk to here? You could better understand what you're doing. You could potentially expand upon, right? And get better results. So yes.
Would our students be receptive to a piece of advice from the old man in the sea?
Absolutely. Well, we appreciate you being here, and we absolutely love any feedback from anybody.
I've spent the better part of 35 years on this campus, and today was a great example of, I see it all the time, giving presentations. If have a 7 to 8 minute time slot, show up with 7 to 8 minutes of slides, 7 to 8 minute total slides. Plan on about a minute per slide, because at this point, the master's, the PhD level, you're almost always going to have slides in there that are pretty complex graphs, and you've got to be able to explain it, because you've seen the graphs hundreds of times. You put the presentation together, you know what's on there. The audience, that's the first time they've seen it. And if you go through them rapid fire, you've lost them. Plan on about a minute per slide. There's some slides you can just go up there real quick, the acknowledgements, the introduction, you can fly right through those. But the meat of your presentation, plan on about a minute per slide, because you've got to get your point across. Even if it's a lot of text on there, don't just stand there and read it. Have the text up there for the audience to be able to read, but provide them with enough information to get the point you want to make across. So that's I see it a lot. Especially the great example of today, you got 7 minutes, and a lot of them you had to go up there and get the hook. You got to go. So, but no, you guys did a great job today. I appreciate the opportunity to be here. But yeah, when I first started here on this campus, there were probably 5 buildings, the entire place. That was a long time ago.
Thank you so much for coming, and absolutely your advice is very good. Considering that a lot of you are just starting your research, it's quite all right to just introduce the field you're looking into, going into, and maybe just present on the why. The why are you doing this, and what, how you might be doing it. Start with that, and then just take one piece at a time for a 7-8 minutes talk. Do it justice, right? That you want somebody to leave with that message, not wanting to just a bunch of graphs that you don't even have time to explain then what purpose that have there. So anyway, citing your, acknowledging your sources for funding is extremely important too, right? Like if you Your project is funded by a funding agency, make sure to put their logo there and to acknowledge them. Other resources, if like, I think I've seen one presentation, but I didn't pay attention. All of them that had the NRT logo, yay. The NSF logo, yay. The GLO, if your project is funded by GLO, you should have it there, or any other funding agency, okay? Right. We'll go, no criticism, this is no criticism. But I think we have a lot to learn from each other too. And so looking forward to your next one. Thank you. Thank you, everyone online. Thank you. No.
I like your outfit. It's so cute. I was like, I really like the top, and then I saw the shoes. I really like it, yeah. It's really pretty.
Thank you.